Distinguishing between primary, secondary, and tertiary alcohols is a fundamental task in organic chemistry. While they share the hydroxyl group, their reactivity varies significantly. Today, we will explore the Victor Meyer test, a colorimetric method used to identify these three classes with precision. The Victor Meyer test relies on a series of chemical transformations that convert alcohols into nitro compounds. The final color of the solution after adding an alkali provides a definitive visual confirmation of the alcohol's original structure. Let's break down the chemistry step by step. In the first step, the alcohol is treated with phosphorus and iodine. This reaction replaces the hydroxyl group with an iodine atom, converting the alcohol into its corresponding alkyl iodide. This prepares the molecule for the subsequent nitration step. Next, we add silver nitrite to the alkyl iodide. A substitution reaction occurs where the iodine atom is replaced by a nitro group, forming a nitroalkane. Notice the formation of silver iodide as a byproduct, which indicates the reaction is proceeding correctly. The third step involves treating the nitroalkane with nitrous acid, typically generated in situ from sodium nitrite and hydrochloric acid. This is the critical branching point where primary, secondary, and tertiary structures react differently due to the number of alpha hydrogens available. Primary alcohols have 2 alpha hydrogens. When their nitroalkane derivative reacts with nitrous acid, it loses water to form nitrolic acid. This molecule contains both a nitro group and an oxime group, which is essential for the final color change. Secondary alcohols possess only 1 alpha hydrogen. Reaction with nitrous acid results in the formation of pseudonitrole. Unlike nitrolic acid, pseudonitrole cannot form a salt with alkali, but it is inherently blue in organic solvents. Tertiary alcohols are the simplest to identify in this test. Because the nitroalkane derivative has no alpha hydrogens, it cannot react with nitrous acid at all. The solution remains unchanged and colorless throughout the remainder of the procedure. The final step is the addition of an aqueous sodium or potassium hydroxide solution. This alkali reacts with the products formed in the previous step to produce the characteristic colors we use for identification. Let's examine the results for each type. For primary alcohols, the nitrolic acid reacts with the base to form a soluble salt called sodium nitrolate. This salt yields a distinct, intense blood red color. If you see red, you started with a primary alcohol. In the case of secondary alcohols, the pseudonitrole does not react with the alkali to form a salt. However, the pseudonitrole itself remains blue. The presence of a blue color after adding the base confirms the presence of a secondary alcohol. Finally, because tertiary alcohols did not react with nitrous acid, adding alkali produces no further change. The solution remains completely colorless. This lack of color is the definitive signature of a tertiary alcohol in the Victor Meyer test. Let's apply this to ethanol. Ethanol is a primary alcohol. Following the steps, it converts to nitroethane, then to nitrolic acid, and finally to a red sodium salt. The red color confirms ethanol is primary. Now consider isopropanol. As a secondary alcohol, it follows the secondary pathway. After treatment with nitrous acid and alkali, the solution turns blue due to the formation of pseudonitrole, identifying it as secondary. Lastly, tert-butanol. This tertiary alcohol forms a nitroalkane with no alpha hydrogens. It stays colorless when nitrous acid and alkali are added, proving its tertiary nature. This consistent logic makes the test highly reliable. To summarize, remember the mnemonic RBC. Red for primary, blue for secondary, and colorless for tertiary. This sequence corresponds to the decreasing number of alpha hydrogens available for reaction with nitrous acid. Think about our summary. If an unknown alcohol yields a blue color in the Victor Meyer test, what is its classification? If you chose secondary, you are correct. The blue color comes from the pseudonitrole intermediate. The Victor Meyer test is a powerful alternative to the Lucas test, providing clear visual results through color rather than cloudiness. Mastering these tests is key to identifying organic compounds. Generated by Vision Solve AI.